Hello dear students. In today's class we will discuss about the concepts underlying Uricotelism, which is a type of nitrogenous waste excretion from the body. So what is basically Uricotelism? It is a process of elimination of nitrogenous waste principally in the form of uric acid. Those animals which exhibit uricotelism are called as uricotelic animals. So in those animals in which the principal nitrogenous based is uric acid are called as uricotelic animals, and the process is called as uricotelism. And where is this uric acid formed from? It is formed in two ways. One, it is produced from ammonia, and second, by the metabolism of purines. Purines are part of nucleic acids. So there are two ways, there are two sources from which this uric acid is formed in the body of uricotelic organisms. They are, first one is ammonia and the second one the purines. Which are the purines? Adenine and guanine. Now, which organisms are basically uricotelic in nature? The birds, lizards, snakes, marine turtles, terrestrial insects, land snails, etc. are primarily uricotelic organisms. And what are the properties of uric acid when you compare it with ammonia and urea? We know that apart from uric acid, the other two main types of nitrogenous waste products which you see in organisms are the ammonia as well as urea, and how we can compare them with this uric acid. Compared to ammonia and urea, uric acid is least toxic. We can say it is almost non-toxic. So even if it is stored in the body of the organism for a long time, there is no problem because it is very less toxic, or it is almost non-toxic when compared to ammonia, which facilitates its storage for longer time in the body. So whenever there is a chance to eliminate it, it need to be eliminated. It can be stored in the body without any threat posed of toxicity. Now, the second is, compared to ammonia and urea, it is relatively insoluble. So it is water insoluble, and it is stored, and because of this water insolubility, it is usually eliminated in the form of either a colloidal solution or a crystal or a paste. So only less amount of water will be utilized for eliminating uric acid from the body. So this minimizes water loss. So what organisms will be resorting to uricotelism? Those organisms which are in need of water conservation, that is the reptiles, birds and the desert organisms. So they mainly try to eliminate uric acid as the principal nitrogenous waste product because its excretion demands only very less amount of water. So it easily precipitates in the form of a crystal, or it forms a colloidal solution. It can precipitate from a supersaturated colloidal solution. So it is excreted in the form of a dry crystalline pellet with little or no loss of water. So uricotelism is a mechanism which will minimize water loss from the body of organisms. And you must have seen the droppings of lizards and birds. They contain white and brownish black materials. The brownish material is their feces, and the white material consists of uric acid crystals, and these are their nitrogenous waste. The brownish black material is the fecal matter. Now coming to the formation of uric acid. So as I already told you, uric acid is formed from two different sources. One is purine and the second one is ammonia. So first we go to purine metabolism. Uric acid is the end product of purines. So where are the purines coming in our body? One, from our dietary sources. From our diet we get some purines. Second is by cellular metabolism. So purines are derived from exogenous as well as endogenous sources. Exogenous means through our diet. Endogenous means the metabolism which is taking place in our cell. So they are contributing to the purine pool. And when we eat proteins, animal proteins, you get purines. Now, The endogenous production of uric acid is mainly from the liver, intestine and other tissues like muscles, kidneys and the vascular endothelium. So now coming to this purine metabolism. So here you can see the two purines, that is guanine and adenine. So we will take this way. By the deamination of guanine, we get a compound called as xanthine. It is this xanthine which is oxidized to uric acid, and the enzyme that catalyzes this reaction is called as xanthine oxidase. So this is considered to be the key enzyme which is involved in uric acid formation. So guanine is deaminated to form xanthine, which is oxidized to form uric acid. Similarly, by stepwise deamination, dephosphorylation of adenine, it is converted to a compound called inosine, which is then converted to hypoxanthine, and this hypoxanthine is also oxidized by xanthine oxidase to form xanthine, which will get converted to uric acid. So here you can see guanine converted to xanthine, which is converted to uric acid. Here you can see adenine is first converted to hypoxanthine, which is then converted to xanthine and then converted to uric acid. So this much of metabolism takes place in our body also. In the human body also this much of metabolism is taking place. Though we are principally organisms eliminating urea, in our body there will be formation of some amount of uric acid also. Okay. Now, in the case of insects, reptiles, etc., this uric acid is further converted to a compound which is called as allantoin. So here you can see. And this is catalyzed by an enzyme which is called as uricase. So uric acid is converted to allantoin by uricase in reptiles, insects, etc. Whereas in humans, this oxidation is not possible because we don't have the uricase enzyme. So in our body, once it is converted to uric acid, it will be eliminated in the form of uric acid itself. But in reptiles and the insects, the uric acid is further converted into allantoin with the help of uricase. Whereas in many birds such as cormorants, pelicans, etc., this uric acid is further converted to a phosphorus rich fecal matter which is called as guano. And guano is very famous as a fertilizer. So in reptiles and birds, this uric acid is converted to reptiles and insects, the uric acid is converted to allantoin, whereas in some birds it is converted to a substance called as guano. So this is the basic metabolic pathway by which uric acid is formed from the purines, guanine and adenine. Now, from ammonia, the second source is ammonia. In reptiles, birds and insects, the ammonia formed by the breakdown of proteins is transformed to ornithine through the ornithine cycle, which takes place in the liver, and then this ornithine is finally converted to uric acid. But this is a complex metabolic pathway and so it is energetically costly. So, when you compare uricotelism with the other modes of excretion such as ammonotelism, uricotelism, etc., what is the advantage? It is that animals resorting to uricotelism will be able to conserve water. They need not spend much of water for the elimination of nitrogenous waste because nitrogenous waste is eliminated in the form of uric acid, either in the form of a paste or a crystal with least expenditure of water. And the second advantage is that this is very less toxic. So whenever there is availability of water, it will be eliminated. You take the case of the bird eggs. The embryo will be growing inside the egg and the uric acid crystals, which is formed as the nitrogenous waste in the embryo's body, will be accumulated in the shell. And it comes out only during hatching when the shell breaks open. So that much time the uric acid is there crystallized onto the shell, but still it is posing no threat to the development of the embryo because it is non-toxic. You take the case of cockroaches, they are insects where also the nitrogenous waste product, principal waste product is uric acid. There it gets accumulated to their exoskeleton. And when there is molting, when there is shedding of exoskeleton, this uric acid is also discarded with the exoskeleton. So that much time it is there in the body. but still it is not posing any toxic effects. So that is the second advantage of uric acid elimination. First is conservation of water. Second is the its non-toxic nature. But the problem is, when you compare it with ammonia and urea, the conversion of nitrogenous waste to uric acid is multi-stepped and it is energetically very expensive when you compare it with ammonotelism and uricotelism. Those organisms resorting to uricotelism have to spend more energy for converting these nitrogenous waste products into uric acid. So it is energetically not very economical, but still what they want more is water conservation. So they go for uric acid elimination. I told you about the formation of some amount of uric acid in humans also. Even though in humans urea is the principal nitrogenous waste product, there is some amount of uric acid also formed from purine metabolism, and this is also eliminated from the kidney through urine. This uric acid is eliminated from the kidney through urine itself. But sometimes when there is elevated level of uric acid, it will accumulate. When there is elevated level of uric acid in the blood, there is a tendency of it to accumulate. It will crystallize and the crystals get deposited in the joints, tendons and surrounding tissues. And this condition is called as gout. And this occurs due to a combination of dietary as well as genetic factors, but this is basically the accumulation of uric acid crystals in our joints, tendons, etc. So this is very much related to our lifestyle, the type of diet we have, and also certain genetic factors. Just take a detailed look at guano, which is the accumulated excreta of seabirds, bats, etc. As I already mentioned, in birds like cormorants, pelicans, and in certain bats, the uric acid is excreted as a phosphorus-rich fecal matter called as guano. So we go to the bird guano. It has got high levels of nutrients, which contains 8 to 21% nitrogen, and this nitrogen content is contributed by about 80% uric acid, 10% protein, 7% ammonia and 0.5% nitrate. So here you can see the nitrogen contribution of guano is mostly from uric acid. 80% of uric acid contributes to the nitrogen content of bird guano, and it also contains phosphorus, calcium, magnesium, etc. So the uric acid gets added on to their fecal matter, and that is what is called as bird guano. It is the dropping of seabirds. And which bird? The bird guani cormorant is historically the most abundant and important producer of guano. And some insectivorous bats also eliminate guano. So this guano, when freshly excreted, it will contain fine particles of insect exoskeleton, which consists of chitin. Because these bats are insectivorous, they feed on insects, and the insect exoskeleton is made up of chitin. So the guano eliminated by the bats will also contain more amount of these particles, and they also contain carbon, nitrogen, sulfur and phosphorus. But here the bird guano, compared to the insectivorous bat guano, the bird guano is having more of uric acid. Now, so with this we have come to the three types of excretion, that is ammonotelism, uricotelism and uricotelism. Now there are some other methods of excretion also. You take the case of some mollusks like unio, limnia, and some echinoderms like starfish, that is asterias. So in such invertebrates, the excess amino acids in the body will be eliminated as such as the nitrogenous waste. That is called as ammonotelism. So the elimination of nitrogenous waste in the form of amino acids is called as ammonotelism. Without much metabolism, the excess amino acids will be eliminated from their body as such, without further degrading them. That is called as ammonotelism, and that is seen in some mollusks like unio, limnia, and in some echinoderms like asterias. In spiders, they eliminate guanine as such without further degradation. Guanine, you know, is a purine. So spiders eliminate guanine as such without further degradation. That is called as guanotelism. So apart from ammonotelism, uricotelism and Uricotelism, there is aminotelism as well as guanotelism. When amino acids are excreted as such, it is aminotelism. When guanine is excreted as such without further degradation, it is called as guanotelism. Now we will go to a phenomenon which is called as dual excretion. That means when animals perform two modes of excretion, it is called as dual excretion. You take the case of earthworms. In rainy days, that is when sufficient water is available, they will show ammonotelism. Whereas when it is a sunny day, that means when there is not enough water available, they will eliminate urea. So they can show both ammonotelism and uricotelism depending upon the surrounding environment. In a rainy day, they are seen to be ammonotelic, whereas on a sunny day, they are seen to be uricotelic. So that is what is called dual excretion. You cannot say them exclusively ammonotelic or exclusively uricotelic. They show both types. Now, lungfishes. Then the xenopus or the African toad, it's an amphibian, as well as the lungfishes. When they are in water, they are ammonotelic because plenty of water is available. We know that for elimination of ammonia, what requires is plentiful of water. So when they are in water, they will be ammonotelic. But during estivation or summer sleep, when they lie immobile in mud or moist air during the summer season, especially when they undergo the summer sleep or estivation, they become uricotelic. So in their life, they are seen as both ammonotelic as well as uricotelic according to the surrounding environmental conditions. So that is also an example of dual excretion. Then the amphibian tadpoles, they are aquatic and so they are ammonotelic. But once they metamorphose into adults, they become uricotelic. The frogs are uricotelic, but the tadpoles are ammonotelic. So in one life cycle itself, you can see ammonotelic mode of excretion as well as uricotelic mode of excretion. Now come to the crocodiles. When in water, the crocodiles are ammonotelic. When they are on land, they are uricotelic. So this is also an example of dual excretion. Now the chelonians. Chelonians are the turtles, tortoises, etc. and the sphenodon, which is a reptile. There you can see uricotelisim in water and uricotelism on land. So when they are in water, they are uricotelic. But once they come to land for egg laying, etc., they are uricotelic. So these are all examples of dual excretion. Now you take man. As it was already mentioned, he is basically uricotelic, but his excreta also, that means his nitrogenous, his urine will also contain uric acid. But you cannot call it dual excretion, but it is just mentioned that along with urea, he will be eliminating small amounts of uric acid also. Then, apart from urea, ammonia, uric acid, etc., there are other nitrogenous wastes also eliminated from the human body. They are creatine and creatinine. They are formed from muscle metabolism. Allantoin, as I already mentioned, produced from uric acid in birds, reptiles, insects, etc. Then trimethylamine oxide, this is a waste in marine teleost fishes or TMAO it is called. Allantoic acid is seen in some teleost fishes. It is a degradation product of allantoin. Then hippuric acid is a waste product in mammals. It is formed when the dietary benzoic acid, the benzoic acid which we obtain through our diet, when it is combined with glycine, it forms hippuric acid. It's a less toxic, it is a less toxic product. So combination of dietary benzoic acid plus glycine gives hippuric acid. It is seen in mammals. Ornithuric acid is eliminated by birds. It is formed by the combination of dietary benzoic acid with ornithine. So benzoic acid plus glycine gives hippuric acid, that is seen in mammals. Benzoic acid plus the amino acid ornithine gives ornithuric acid, which is eliminated by birds. So these are the different types of nitrogenous excretion. Thank you.